Morning, everybody. I think we'll go ahead and get started. Big thing is that for Monday, we have exam number 3. So Monday morning here, right there, I'll be in at 7:30 a.m. If you guys want some extra time to work through that exam, extra time to take that. But we'll start that on Monday morning. That does cover the community ecology lecture set, the ecosystem, energy flow and nutrient cycling through ecosystems lecture set, the ecosystem ecology lecture set, and then the most recent conservation biology lecture set. So that is what's going to be covered on that particular exam. So we'll do that Monday. Any questions or anything you guys want me to maybe talk about a little bit before I kind of go into the lecture material for today, which is going to be exam 4 content? Yeah, go on.
And where like Adam, we maybe can like remember.
So typically the terrestrial biomes, one of the things to kind of keep in mind, yes, is where they're at typically in terms of Earth's areas. And so some of them are more equatorial or sub-equatorial, like the tropical rainforests or savannas and things like that. Those are going to be more towards certain equatorial regions. The deserts are typically, a lot of times deserts around the 30 degrees south or 30 degree north latitudes. But you do move up through, as you move up through the latitudes from the equator, either north or south, you get different types of biomes. And yes, you might need to know roughly where some of these biomes typically are. But that's so, I don't know if there's like a way to remember that other than just to make sure you're just kind of studying through them.
More precipitation or more nutrients or less.
Typically, the cooler the ecosystem, the more it's away from the equator. And so that can actually kind of help. So like temperate broadleaf forests, those have have pretty cold winters, but conifer forests are even colder over winter and stuff like that. So you can kind of actually, that just the climate conditions, the weather conditions are also directly related also to their latitudes then as well. So there's some things that go hand in hand, and because those are some patterns that can help you remember some of them then too. Other questions? So the exam, if you guys do have any questions, feel free to interrupt me anytime today. The exam's on Monday. Today, though, lecture is going to be on exam 4 content, so the next one. So exam's on Monday. Tuesday, we are going to have a lab on protistans. We're going to start talking about those. We're going to talk about those in lecture as well. But there is a pre-lab quiz to do before you come to lab class on Tuesday then. Next week, there will not be a bright space quiz since it's an exam as well. So next week's just exam 3 on Monday and a pre-lab quiz on Tuesday, and that's the only things for next week. With that being said, again, if you guys think of a question as we're going through lecture today that you want to ask over exam 3 content, then please go ahead and feel free to kind of interrupt me. But otherwise, we're going to move on just so that we're not rushing through the material on to talking a little about the next topic in class, which is prokaryotes. And this is going to be exam 4 content. So far to date in biology 2, we've talked about things like Mendelian versus non-Mendelian genetics, evolution, population genetics, phylogenetics, taxonomy, species and speciation, and we've kind of gone through our ecology unit and talking about ecology at different levels like population, community, and ecosystem ecology. This next kind of unit in the class and exam 4 content is going to be a little bit different. but we're not going to be talking about some specific kind of groups of organisms found in nature. We're really going to be talking a little bit about biodiversity. What are some main groupings of organisms? But we are going to be talking about their ecology and the evolution of some of these types of organisms then as well. So some of the topics we're going to talk about earlier in the class are going to come back up, but we're going to apply them to kind of specific groupings of microorganisms. Now, we're going to be mostly focusing on microorganisms, types of microorganisms for the remainder of the semester. When I say microorganisms, what does that term mean? Yeah, so they're going to be tiny. They're going to be things that we cannot see with the naked eye. So microorganisms, this is any organism too small to be observed by the unaided eye. How do we actually observe them instead? What do we use? Yeah, microscopes. And so we use microscopes to kind of see these, but we can't see them with our eyes every day. What are maybe some examples of microorganisms? So bacteria is going to be a main grouping of microorganisms. Or maybe some others. Enzymes are not going to be microorganisms. They are too small to be seen, but they're not living organisms. So enzymes by themselves can't reproduce, for instance, or have those characters. Particularly talking about enzymes are just part of cells, but we are still talking about cellular organisms. Protists, many protists are microorganisms, but not all, which is something we're going to talk a little bit about. The archaea. And we've got a few. So what are some different types of microbes? The bacteria, and that's some of the ones that we're going to be talking about. The archaea. And we're going to be talking a little bit about those in the next couple lectures. Bacteria, archaea are going to be the focus for our next couple lectures. But there are things like the protists. You guys did a pretty good job of naming some of these so far. But then there's also things like the fungi. Many species of fungi are microscopic. For instance, yeasts is a general term for any type of unicellular fungus. And so many fungi are then microbes. We have sometimes even micro animals, for instance. So there's some types of worms that are microorganisms are too small to see. Things like parasitic worms, like tapeworms at parts of their life cycle are too large to be seen, or roundworms or pinworms. There's also some things like nematode worms. And you guys might not know it, but in the summertime, you probably are stepping on about a good 100,000 nematode worms sometimes when you're going through some wet soil areas. They're all over in the ground there, and we can't see them at any given moment. So there are some microorganisms, even animals, and these microscopic worms, for instance. And then sometimes the viruses also get lumped into this category then as well. So these are some different types of microbes, and this is going to kind of be our focus over the remainder of the semester. We're going to be talking about these different kinds of microbes, but then also what are the kind of hallmark characteristics of some of these microbes? What are some of their ecological functions, and how have they evolved and changed over time? That's really what we're going to be talking with some of these. Now, when we look at all these different types of microbes, I also have shown you this diagram before. This is the tree of life. It shows the evolutionary relatedness of organisms, and it divides them up into the 3 domains. What are the 3 domains of life? So it's the eukarya, the bacteria, the archaea. So we have the bacteria, the archaea are here in red, and then all of the eukarya are in yellow. So this is, again, just shows the evolutionary tree of life. It just basically sees, if I trace back all the bacteria to this branching point, what does this branching point on my diagram, my tree here represent? Yeah, the last common ancestor for all bacteria. And this would be the last common ancestor for all archaea. This would be the last common ancestor for all eukarya. Why are they divided up into 3 domains? Because this is a tree that is a hypothesis. It's an evolutionary hypothesis. We talked about what trees were earlier, but it is based upon data. things like 16S rRNA phylogenetics and other forms of data. So this is a data-driven tree, but what it shows is our evidence shows that all the bacteria can be traced back to a common ancestor before you hit any member of the archaea of the eukarya. And that means all the bacteria are more related to one another than any member of the other domains. Does that make sense, everybody? Same thing, all the archaea can be traced back to one single common ancestor. All the eukarya can be traced back to one single common ancestor. And so our evidence suggests they are these groupings or these domains. And that's why we're doing them into these. Now, when we look at these domains, what we've also found is a lot of the eukarya are fairly closely related to them. So the eukarya includes all plants. So that is here on this, all animals, all fungi, and the rest of this stuff that you're seeing up here are different kinds of protist species. We're going to talk a little bit about now also the subdivisions in some of these different domains then as well. Now, the one thing I want to point out though, if we look at this tree, most of the tree is microorganisms. So the vast majority of species on the planet is going to be microorganisms and microbiology. All of the bacteria, all of the archaea, the viruses, most of even the eukaryotes are then microorganisms. And most of them are unicellular, for instance. So we're going to be talking a little bit about this. Typically, the only things that are not kind of microorganisms are plants, animals, some fungi, and some protists are sometimes more macroorganisms, things that we can see with the onion. The other thing, if we're looking at these trees, is all of the eukarya are eukaryotes or prokaryotes. Eukarya, all eukarya are eukaryotic cell types. But what's that probably then mean about the bacteria in the archaea? Yeah, the bacteria in the archaea are going to be prokaryotic cell types. So bacteria are prokaryotes, archaea are prokaryotes. And what we're going to start talking about today is the prokaryotes. We're going to talk about both the bacteria and the archaea in this particular unit. The rest of the semester is then we're going to talk about, after that, we're going to talk about protists, we're going to talk about fungi, and we're going to talk about viruses. Are going to have their own little units then as well. So prokaryotes, these are amongst the most abundant, most successful, and also most diverse set of organisms on Earth. What I just mean by that is there's a lot of different kinds of prokaryotic species doing a lot of different things. So there's lots of prokaryotic species filling lots of different niches or ecological roles out in the environment. So they have very diverse roles, very diverse adaptations that help them succeed in different areas. But we can have bacteria or archaea. Sometimes we can have them under kind of normal conditions, like out in water and soil. We can have them in hot spring that go up to boiling temperatures. We even have some archaea that lived at temperatures at 113 degrees Celsius in some cases. By the way, what's boiling? 212 Fahrenheit. What's boiling in Celsius? 100. So there are some archaea species that live above boiling temperatures on a regular basis. You can find these things, you can find these floating up in the high stratosphere, nearly up to space. So you can find these guys just about anywhere and everywhere. Again, with the prokaryotes though, they are divided up into the two domains of bacteria and archaea. The bacteria are prokaryotic, the archaea are prokaryotic. We're going to talk a little bit about what it means to be a prokaryote versus a eukaryote. But then we're also going to differentiate what are some structural and physical differences between the two bacteria prokaryotes, which are the bacteria and archaea then as well. The first thing, what is a prokaryote versus a eukaryote? You guys have had this a couple times already. Hopefully this is the easy part. Yeah, go on. Oh, sorry, no. It's okay. So with the prokaryotes, typically if we look at eukaryotes versus prokaryotes, if we're really trying to define the difference between these, it's actually not the easiest thing to define. So we tend to focus on what the differences are between a eukaryotic cell versus a prokaryotic cell. So what I have here up is a diagram. This is a kind of maybe typical generic eukaryotic cell. This is then a prokaryotic cell relative to it in size. On average, most prokaryotic cells are about 0.5 to 5 micrometers. That's much, much smaller than about the 10 to 100 micrometers in diameter that these eukaryotic cells. This actually makes prokaryotes 1,000 times less volume on average than eukaryote in essence. So typically prokaryote cells, this is my prokaryotic cell. This is just blown up a little bit so we can take a look at some of the inside stuff. But this is basically a prokaryotic cell on average size relative to then a eukaryotic cell. Prokaryotic cells are typically, not always, but typically much smaller than eukaryotic cells. The other thing is that eukaryotic cells have membrane-bound organelles, but prokaryotes don't. So prokaryotes lack organelles. What's an organelle? So what are some examples of organelles? So a mitochondria might be a organelle. So eukaryotes would have mitochondria, for instance, but prokaryotes do not have mitochondria. What were maybe some other examples of an organelle? Golgi apparatus. That's another example. We could have chloroplasts. We could have ER, endoplasmic reticulum. We can have some different types of organelles. What, though, is kind of the definition of that term, organelle? Yes, sort of. You are very close. So they are going to be, they're going to have, organelles are structures that have a function, a specialized function in type of cells. Anything else that's particular about organelles that they always have? There's substructures inside the cell, but how do they separate it from the rest of the cell? Yeah, so organelles are going to be membrane bounded compartments of specialized function. And so what they do is there are these membrane bounded or membrane contained subcompartments inside the cell, but then they're going to have some type of specialized function then for the cell as well. So for instance, what's the specialized function for a mitochondria? It's energy processing, and it does part of cellular respiration. It does the citric acid cycle, or sometimes it's called the Krebs cycle, plus it does respiration, and then it does oxidative phosphorylation. It doesn't do glycolysis, though, which is traditionally part of cellular respiration as well, when we describe that. Cellular glycolysis is in the cytoplasm. So we have, so it does things like part of cellular respiration. What's the specialized function of chloroplasts? Photosynthesis, although specifically the light reactions of photosynthesis, not necessarily. So we have these different organelles that have these different specialized functions. So we might be doing some things like that. Is the nucleus an organelle? Yes. And the nucleus' specialized function is anything related to DNA, in essence. Now, all eukaryotes have organelles, prokaryotes do not have organelles. Closely related to that, nucleus is an example of an organelle, but all eukaryotic cells have a nucleus. This is a membrane enclosed compartment. Again, that contains the DNA of eukaryotic cells, but also any function related to DNA, like DNA replication, Or transcription, where DNA is used as a template to make what? RNA. So all of that's gonna take place inside of the nucleus of eukaryotes. But again, prokaryotes don't have organelles and they don't have a nucleus. So the nucleus is a membrane closed compartment that contains the DNA of eukaryotic cells. Prokaryotes don't have a nucleus. Instead, they have a structure called a nucleoid. Their DNA is just in the cytoplasm. There's no membrane around it, but the nucleoid is still where that DNA is wrapped around some structural proteins, and it still is a organized structure, even though it's not membrane bound. So the nucleoid is really just DNA wrapped around proteins into a organized structure in the cytoplasm. So it's not membrane bound, it's just going to be free floating in the cytoplasm as well. All right, there are some other differences though as well between a eukaryotic versus a prokaryotic cell. So for instance, eukaryotes, most of the time, if we look at their DNA structure, eukaryotes, most of the time they have multiple chromosomes, and those chromosomes then are linear. So they might have 1, 2, 3, 4 chromosomes, so they might have 4 different, and those chromosomes have ends. So they're linear DNA molecules. So multiple chromosomes, but also then linear chromosomes where they're going to have ends. Typically then most prokaryotes only have 1 chromosome, and that chromosome is a circular DNA molecule where the DNA doesn't have ends, instead it's one big long circular DNA molecule. So that's going to be a key difference. There's always some exceptions to these rules, by the way. But typically, most of the time, eukaryotes have multiple chromosomes, and those chromosomes are linear in nature. And then prokaryotes typically have only a single chromosome, and they are typically circular DNA molecules then. Ribosomes are also different. Eukaryotes have what are called 80S ribosomes, and then prokaryotes have 70S ribosomes. 80S versus 70S, this is a way, kind of an old school way of measuring mass of the ribosomes. And really what this is just saying is the 80S ribosomes, they're bigger. They have a little bit extra mass, they have a little bit extra parts. But the 70S ribosomes are a little smaller and have a little less mass. But there are some structural differences between them. But that being said, what do ribosomes always do? Yeah, ribosomes, the function of ribosomes is to make proteins, to synthesize proteins then. But they do have different ribosomes, and they do have different 16S rRNA sequences. So we talked a little bit about 16S rRNA phylogenetics earlier in the semester. But they also have different, noticeably different 16S rRNA sequences as well. They have different sizes, different parts, different 16S rRNA. Another difference between prokaryotes and eukaryotes is sometimes their cell wall composition. Now, this is a little complicated though in eukaryotes. There's a bunch of different kinds of eukaryotes. And different eukaryotes have different cell walls around them, and some of them don't have cell walls around them. So for instance, animal cells, do we have a cell wall around our cells? No. So something like animal cells don't have a cell wall at all. But plants, what's the cell wall of plants? Cellulose. And then something like fungi cells have a cell wall made out of a substance called chitin then as well. So different eukaryotes do different things for their cell walls. That's not really what the difference here is. The difference is actually that prokaryotes use specific cell walls. So bacterial cells specifically use a substance called peptidoglycan as their cell wall. We're going to talk in detail about what peptidoglycan is and peptidoglycan's function and structure. But bacteria, almost all bacteria have a cell wall made out of peptidoglycan. Archaea, you notice I didn't lump onto here. Archaea have a cell wall made out of a substance called pseudo-peptidoglycan. What's pseudo mean? Fake or false in Latin. So it's basically their cell wall is made out of something called false peptidoglycan. It's a molecule that's similar to peptidoglycan, but it is structurally distinct to it. So all bacteria have cell walls of peptidoglycan. Archaea have cell walls of pseudo-peptidoglycan. These are similar, but they're not the same thing. So that would be a difference between, say, bacteria and archaea. All right, questions about eukaryotes versus prokaryotes. Be careful with the cell wall one. This one is not wall versus no wall. It is nuanced, and the details matter. Especially because we're going to talk about things like fungi, and you're going to need to know later on that they have a chitin cell wall. We're going to talk all about fungi here in a later unit. Okay, so bacterial structure. We're going to look at a little bit at the overall kind of body plan or structure of bacterial cells, and then we're going to look at the overall kind of structure of archaea cells and some differences there then as well. So with this prokaryotic body plan, we're just going to be looking a little bit at the cellular boundaries, things like the membranes and the cell wall. We'll be looking at a little bit of structures inside the cell in the cytoplasm, and then also maybe some structures, external structures like flagella that are sometimes involved in things like cell motility, or a few other external structures that might be involved in a few other types of processes. First off, the prokaryotic cell shape. Probably the three most common shapes when it comes to prokaryotic cells are cocci, which are spheres. So a cocci is a spherical cell. By the way, coccus is singular, cocci is plural. These are all Latin derived and use kind of the Latin singular and Latin plurals. Bacilli is then, or bacillus again. Bacillus is singular versus bacilli is plural. But a bacillus-shaped cell is going to be more of a then rod shape, or kind of think of it as more rectangular, but more of a barrel or a barrel shape. And then we have spiral-shaped cells, or sometimes what's also referred to as spirillum-shaped cells as well. So these are kind of the three most common shapes when it comes to prokaryotes. And most prokaryotes come in one of those three shapes. There's also some other shapes that are less common, like for instance, vibrio or prosthecate or pleomorphic. Don't worry about those too much. These are the three most common, and most bacteria archaea come in one of these three shapes. If we then look at the shape of the cells versus now, maybe we look at some structures that are on the interior of the cell. And when I say the interior, I mean they are in the cytoplasm. Okay, so when we're looking at the cellular interior, we're going to be looking at structures or things that are in the inside of the cell, the cytoplasm of the cell. So all the stuff I'm going to go through in this next unit is in the cytoplasm. First thing is just the cytoplasm itself. Cytoplasm is about 70% water on average in cells. That being said, in the textbook, you guys see the cytoplasm in the textbook often looks like this nice empty space, right? Not a lot going on there. That's not the way real cytoplasm is. Cytoplasm instead looks like this. It is completely chock full of stuff, and there's very little empty or free space. And there's very little free water. The water is constantly hydrogen bonding and interacting with everything else in there. So what you have is that cytoplasm is completely full of a wide variety of proteins, of DNA, of metabolites, of water, and all that stuff is constantly interacting with one another in this soup. Although I call it a soup, it's really almost more the consistency of molasses If you could get cytoplasm on your fingers, it would be really thick, viscous liquid. And syrupy, molassesy in terms of its consistency. That's because it's completely full of all this stuff. So this is a prokaryotic cell right here. It's a little diagram. But what is all this different stuff? Well, this stuff right here that you're seeing kind of in this yellowish color, this is DNA getting wrapped around proteins. The DNA gets wrapped around proteins and forms a structured ordered nucleoid. So in essence, this is actually the nucleoid right here, for instance. There's the DNA wrapped around a bunch of proteins. From that DNA, you also have some RNA polymerase in here. And that RNA polymerase is making RNA molecules. And the little white stringy things here are then some RNA molecules that are floating around in the cytoplasm. You have in the purple, these are a bunch of ribosomes. And the ribosomes are then making a bunch of proteins. And the stuff in blue are just the different proteins being made by those ribosomes inside of the cell. So that's kind of what you're seeing in this particular diagram. You're also seeing some other structures, like this is the plasma membrane here, this is the peptidoglycan cell wall, this is the outer membrane, and then this is a flagella. So there's a bunch of stuff going on in this diagram, but it is just showing kind of, I've used this diagram to show the cytoplasm is completely full of all of this stuff in there. And things are constantly bumping into other things. So it's a lot more complicated. And in fact, it's almost impossible to use computer programs to model the inside of cells and what happens inside the cells because there's so much stuff bumping into other stuff all of the time. It's a very difficult thing to do. They can barely model how one protein might act inside of a cell, let alone multiple proteins interacting with like a cell. There's actually projects, there's like, oh, we're gonna put a protein into this solution, how's it gonna interact? And then they use about like 200 hours of computing time just to compute how one protein might fold up. And it still doesn't work much of the time. So that's like computational chemistry types of things involved with that. All right, what else is inside of that cytoplasm? We have nucleoids. Nucleoids, again, we just talked about this, but this is a region of the cell that contains a single circular chromosome of prokaryotes. And that chromosome, that DNA, that chromosome gets packaged up and wrapped around proteins. It is an organized structure. So this is an electron microscope image of a prokaryotic cell, but this region right here, you see that's a little lighter, that's the nucleoid region. So it is, the DNA is packaged and condensed. It is an organized structure inside of there. It is not membrane-bound nucleus. And here you can actually see, again, this is fluorescent microscopy staining, but they use a green stain that stains the membranes and gives off green fluorescent light. They use a stain that binds DNA and then it fluoresces red, so you can visualize where the membranes of the cells are versus where then the DNA of the cell is, for instance, then as well. Now, it is, again, prokaryotes have a single closed circular chromosome. So again, it just looks like this. This is still a double-stranded DNA molecule. I have one of the DNA strands marked in red, the other DNA strand marked in blue, but it is a single closed circular DNA molecule. That is the chromosome of it. Typically, on average, in prokaryotes, the chromosome size is about 0.5 to 10 million nucleotide base pairs long. And when I say the nucleotide base pairs, I mean the A, T, Cs, and Gs. So it's anywhere from about 500,000 to 10 million A, T, Cs, and Gs long. For instance, Escherichia coli has a chromosome size of about 3.5 million base pairs. Certain species of cyanobacteria, photosynthetic bacteria, are more up around the 10 million mark in terms of the number of base pairs. That chromosome then codes for, on average, anywhere from 500 to 10,000 genes then as well. Anybody happen to know how many genes we have in humans?
A billion.
We have about 3 billion nucleotides in our genome. Actually, that's a really good guess. It's estimated between 20 to 25,000 genes in human genome. So we're not sure exactly, but something like bacteria have about 5 to 10,000 genes. We have maybe about 20 to 25,000 genes in our genome then instead. That's a lot of DNA though. This is an E. coli cell, but it's died in an electron micrograph image, and this is the cell broke open. This is its DNA spilling out. And so that is a lot of DNA to fit into a very tiny cell when we're talking about these numbers here. That's why, again, it has to get packaged around proteins. It has to get wrapped, looped, packaged. It has to get packaged around those proteins in order to even fit inside the cell in the first place. Questions about that? If you could stretch out our DNA from end to end, does anybody know how long it could be? No, it's not that impressive. It's microscopic still. It's about 3 meters. Be taller than you, though. It'd be cooler if it wasn't. Stole my butter, Isaac. No. All right. All right. So, question about the inside of the cell. Oh, the other things we have inside the cell, like I said, we have these ribosomes in the cytoplasm. We have the nucleoid. We have a bunch of stuff in there in this one, a bunch of proteins and things like that. Questions about that, though? All right, next we're going to talk a little bit about prokaryotic bacterial cell boundaries. And here we're going to be focusing a little bit more on then the membrane of the cells and the cell wall then as well. So the cellular membrane, this is the main structure that delineates the in versus the outside of a cell. So this is a pretty thin structure. It's only about 6 to 8 nanometers thick. But it is a very vital and then selectively permeable barrier around the cell. What's it mean to be selectively permeable? It only allows certain things in and out of the cell. The membrane, what is most of the membrane composed of? Membranes are mostly what? Phospholipids. And then just one phospholipid or the whole lot of them? And then how are those phospholipids organized? Into a bilayer. Okay, so do you guys remember what a phospholipid is by any chance?
What it looks like?
Or the parts to a phospholipid?
A
A hydrophobic side,
hydrophobic head and.
hydrophilic head. So it is going to have a polar head group. It gets attached to a glycerol molecule. So the polar head group is attached to the glycerol. And then anybody happen to remember what the other part of phospholipids are? Hydrophobic tails. 2 of them also attached onto the glycerol, but these are fatty acids. But they are, so the polar head group is hydrophilic because it is charged. It actually is going to usually have a negative charge because it has phosphates as part of that one, phospholipid phosphate. And then the fatty acids are hydrocarbon chains. And are those going to be charged or uncharged? Hydrocarbons, just carbon. They're made up of a bunch of carbon and hydrogen. Yeah, they're going to be uncharged over here on this side, or again, hydrophobic. Fatty acids are going to be hydrophobic, uncharged. So the phospholipid has a uncharged hydrophobic side, and then it has a charged polar head group side that's hydrophilic. Does that make sense, everybody? But with these phospholipids, if you then have them, if you have many, many phospholipids in a aqueous solution, a water solution, they will spontaneously, all on their own, come into a structure called a phospholipid bilayer. So right here, this right here is one individual phospholipid. Okay, like I just kind of drew out. So these are my fatty acid tails. My squiggly lines in the middle are the fatty acid tails. Those are hydrophobic uncharged regions. The green sphere then is supposed to be my polar head group then as well. And that's going to be a hydrophilic because it's charged. And charged things like to interact with other charged things. Is water charged? Water has partial positive or negative charges on the oxygen. Negative and partial positive charges on the hydrogens. Because why is water partially charged? So this hydrogen and oxygen bond, is that, what kind of bond is that? Is a covalent bond, a hydrogen bond, an ionic bond? It's a covalent bond. What kind of covalent bond is it though? So there's single covalent bonds, double covalent bonds, triple covalent bonds, but there's also polar versus non-polar covalent bonds. So this is going to be a polar covalent bond and a single covalent bond involved here. So polar covalent bonds, what happens is the oxygen, right? The covalent bond is a chemical bond where the two or more atoms share electrons. But the oxygens are a little bit more greedy and they tug the electrons a little closer to themselves. Electrons carry what charge? Negative. But if the oxygens are kind of not sharing the electrons completely equally, the electrons and the negative charges go closer to the oxygen, and it makes the oxygen partially negative because those electrons are moving away from the hydrogens, but you're left with a proton here. These are partially positive because protons have that positive charge then. So that's just what's going on there with a quick review of water. But what happens here is the bilayer has these hydrophobic regions. These are uncharged. These don't like to interact with water. The polar head groups are charged, like dissolves like, charged things like to interact with other charged things. So in this case, the charged polar head groups like to interact with water. If you mix a bunch of phospholipids, they automatically are forming this structure as the most stable. It's called a phospholipid bilayer, where you have one layer of phospholipids and then a second layer of phospholipids. But the fatty acid tails of one layer point to the fatty acid tails of the other layer, and hydrophilic head groups are always on the outer side of these interacting with water. This is going to be naturally the most stable structure. What that does, though, is it sets up this substantial hydrophobic region in the middle of the membrane. That's the thickest part of the membrane. So I also said that the membranes were selectively permeable. What kind of substances can pass through membranes? So oxygen gas can. CO2 gas can. Sodium cannot pass through membranes. Well, what can pass through? These are small, so that's one reason they can pass through membranes, but also they are things like oxygen gas and CO2 gas, they are uncharged. Only small uncharged things can pass through membranes. Charged things like ions, and again, sodium is an ion. Ions and other charged substances can't pass through membranes. Questions about that? Are phospholipids the only component of membranes? Is there any other kind of macromolecule that's common in membranes as well?
Protein.
Yeah, there's also going to be the phospholipid portions to membranes, but there's also going to be protein portions to membranes, but also there's usually some carbohydrate, sugar portions to membranes then as well. So membranes are important to protect the cell. That's going to be their main function. Another thing that's important to protect the cell is the boundary of the cell is peptidoglycan, the peptidoglycan cell wall. Peptidoglycan is a very protective structure. It provides both protection from harmful chemicals, especially provides protection from osmotic damage. What do I mean by osmotic damage? Or what is osmosis?
The movement of.
Yeah. What happens if too much water moves into a cell? Cell can burst open. The peptidoglycan cell wall is a very strong, very rigid structure, and it can keep the cell from bursting open as long as the osmotic conditions are not too strong. So for instance, it can help the prokaryotic cell survive in things like freshwater environments, but maybe not distilled pure water, for instance. If you put a cell into pure distilled water, you're gonna kill it. The water moves from osmosis. How does water move during osmosis?
Well, I can tell you that.
Water moves relative to solute concentrations that are dissolved in the water, and it's always going to move from areas of lower solute concentration to areas of higher solute concentration. Pure water has no solutes in it, very low in essence, and that would cause the water to move into the cell and the cell could burst open. Cell wall helps to protect that from that osmotic deformation. And so it also helps to maintain the shape of the cell. So that's a kind of three different kind of main functions here. Almost all bacteria have these cell walls made out of peptidoglycan. And peptidoglycan is called peptidoglycan for a reason. It has a sugar component. The glycan is just a generic sugar name, but it has a sugar component. So we look at the structure of peptidoglycan. It has two different sugars, N-acetylglucosamine, or NAG for short, and N-acetylmuramic acid, or NAM sugar for short. And what happens is it makes these polysaccharides. What's a polysaccharide? What does poly mean? Many saccharide sugars, many sugars joined together. In this case, it creates a polysaccharide chain of NAG, NAM sugars, but they actually just go like this: NAG, NAM, NAG, NAM, NAG, NAM, NAG, NAM. They alternate. And it builds a polysaccharide sugar chain, many sugars joined together, but it always alternates NAG, NAM, NAG, NAM, NAG, NAM. But you get one polysaccharide chain, you get another, you get another, you get many of these polysaccharide chains formed. That's going to be the sugar component of peptidoglycan, but then it also has a protein component, peptido. Peptido just means peptide bonds or peptides or proteins. Those are terms that are used somewhat interchangeably. The protein component is then that there are some amino acids that act as cross-linkers between one chain to the next. So this is one polysaccharide chain, but then this is going to be an amino acid bridge or cross-linker that will join this polysaccharide chain onto then this second different polysaccharide chain. So what you're seeing here is the peptidoglycan. This is just like if you had the cell and you cut it. This is then the cytoplasm here. This is the, what? This next layer here. It's going to be the membrane, and then outside of that is then the cell wall. So that's what you're kind of looking at here, is the peptidoglycan wall is always outside of the plasma membrane. That as well. Now, this just acts as kind of almost a molecular mesh. So again, this is just a different way of me showing this. But again, it's just repeating units of NAD and NAM sugars to build a polysaccharide chain, a second polysaccharide chain, a third polysaccharide chain. And then those polysaccharide chains are joined by these amino acid cross-linkers to one another. Any pattern you know, and any pattern you guys see with the amino acid cross-linkers? They're always on which sugar? Yeah, they're always on an NAM on one polysaccharide chain. The amino acid cross-linker is always attached to an NAM on one polysaccharide chain and joined into an NAM on a second different polysaccharide chain. It's always the NAM sugars involved in those amino acid cross-linkers. Question about that? Last thing for today, though, and it's just a quick summary, and we're going to talk about this then on Wednesday after the exam. But where we're going to pick up talking is about those two main cell architectures for bacterial cells. Bacterial cells usually are either gram positive or they are gram negative. And it has to do with their architecture. But gram positives, this is the inside or the cytoplasm cell. They have a plasma membrane, and then outside of that, they have a thick peptidoglycan cell wall. and that's a gram-positive cell architecture. Gram-negative architectures are more complicated. Again, this is the inside of the cytoplasm cell. They have then a plasma membrane. They still have a peptidoglycan cell wall, but it's often thinner. But then outside of that, they have a second different outer membrane. And the outer membrane and plasma membranes, they're both membranes, but they're not the same. They have some structural differences. And we're going to talk about what those differences are then as well. But we'll pick back up with that on Wednesday.